I am James Gulley. I'm the Chief of the Genitourinary Malignancies Branch and Director of the Medical Oncology Service. But really, those hats aside, I'm here as a practicing clinician, clinical trialist, and also member of the NIH Ethics Committee. So I do have a little bit of background, and I've been fortunate enough to be involved in this course for a number of years now. And so what we want to do today is really to talk about problems that can arise if we don't really stick to our strong guidelines about scientific integrity, really as it relates to clinical trials and clinical trials process. We're going to make this interactive. We're going to make this kind of a little bit fun by going through some of the cases of people that have run into problems, and hopefully we can use those case scenarios to help us avoid the problems. So I think the first thing we need to focus on is scientific integrity. So first of all, what is scientific integrity? Well, we should all be committed to the process of the responsible use of the scientific method to seek new knowledge. And certainly all staff within the intramural research program should maintain exemplary standards of intellectual honesty in formulating, conducting, and presenting research as befits the leading role of the NIH. So why is scientific integrity so important? Well, I think it's important in part because the scientific community and the general public rightly expects adherence to the standards of intellectual honesty in the formulation, conduct, and reporting of scientific research. Without this high standard of scientific integrity, the scientific community and the general public may become victims of research misconduct. And that's what we're going to talk about in the next few minutes here. So what is research misconduct? Anybody want to take a guess as to what the three areas of research misconduct are? Go ahead, just shout it out. Yeah, falsification, heard that, good. Plagiarism, yeah. And another one that begins with F. Fabrication. Okay, so let's talk. What is fabrication? It's just plain old making up data, okay? And recording or reporting them. Falsification is manipulating data, so maybe not completely made up, but you may actually change some of the data or some of the results, or omit some of the data, and we'll talk about that in a little bit. And then plagiarism. Probably most of us know what plagiarism is. It's basically taking another person's ideas, processes, or words without giving the appropriate credit. So research misconduct does not include an honest error or a difference of opinion. That is different than research misconduct. Also, just within the NIH guidelines, if you are a scientific collaborator of somebody else, taking your words, how you wrote it in one article and using that in another article in a similar way, it probably doesn't rise to a level of plagiarism if it's, for instance, a research article. Okay, now we're going to switch gears here and go actually into some real-life examples of scientific misconduct. So this was a case in 2006. This Korean investigator, Woo-Suk Hwang, who claimed to have created the first ever human embryonic stem cell lines, and the DNA matched that of the patients. And so this was going to create huge new breakthroughs in treatment modalities for patients. So the case, it turned out, involved some questionable manipulation of the data, and we'll show you some of the pictures from that in just a minute. So it turns out that after all was said and done, of the 11 original cell lines, 9 of those were fabricated, and the images of the cells were manipulated on the primary journal. And so one of the results of this particular egregious case was that now virtually all the major journals, they look in their review process at the images, and they look to see, are these images manipulated? And so we're going to show just a few examples of how they can tell that. Oops, sorry. So here you see in the top panel here, this is the original submission, and this was a supplemental figure. So just because it wasn't in the print figures, don't think that this wasn't anything that you submit, even if it doesn't get published, if it's submitted to the paper, that at that point, if it's fabrication or falsification, you are potentially, there's potentially scientific misconduct going on there. So you can see here very dark, very kind of a little bit of light in the middle here. These are two cell lines, and this is a negative. But you can see here, actually, when you adjust the tonal range, you can see that these two figures are identical. And indeed, this was just manipulation of the data here and fabrication, because this was not a separate cell line from this. So this is another interesting case. So this was actually an example of manipulated data, and this was something that was published by one of our researchers here, just showing how potentially one could manipulate data and then look to see if that, how you could uncover whether that data was manipulated. It's actually a very interesting read if you want to read this, and it's called The Temptation of Image Manipulation. And you can see here a very nice picture of these cells here, but when you contrast adjust this, you can see that everything here in the background has this grainy appearance, whereas here it's dark, except right around these cells there's this dark area that's completely black. So you could tell that they cut and pasted this from a different picture, and so that these cells here and here really weren't in the original photomicrograph. Back here. So just one other quick recent issue where there was also image manipulation, and also actually with stem cells. This was a researcher in Japan, and this researcher actually submitted data to Nature, was published, but within days of the publication, data came out that there was images that were manipulated, there were cell lines that were fabricated, and he actually lost his position in the academic university there and lost all the grants that he had, etc. Now moving on and talking about clinical trial data manipulation. So this was data from a breast cancer prevention trial that was funded in part by the National Cancer Institute. A data coordinator actually falsified and fabricated the test results of the exam. So basically, patients came in and there was incomplete data there. All the data wasn't captured. Maybe the patient either didn't come in for a test or they didn't write down when the test was done. And so she just made up the dates so that she could fill in the blanks. Another case in the National Surgical Adjuvant Breast and Bowel Project, a data coordinator falsified and fabricated follow-up data. So these were data when you're supposed to follow the patients every 6 months for X number of years, and maybe all the patients didn't come in, or maybe they didn't report everything, or it wasn't written down, and they just, again, filled in the blanks. So what's so wrong about doing that? Well, certainly inaccurate or misleading data regarding patient follow-up can have big implications, because these studies were designed to see, does a certain intervention impact the clinical course of a patient compared to a different intervention. And you know, if you have wrong data in, you can't look at any of the data within that project and know whether or not there is any difference there. So it just makes the whole study uninterpretable. Now, this is perhaps one of the most egregious cases here that I'm going to go through. A case that really affected many hundreds of patients over almost a decade of treatment. So back in the late 1990s, hematopoietic stem cell transplant for patients with breast cancer was actually a pretty big and growing area of interest. People felt that if giving a little bit of chemotherapy and we're seeing a response, maybe we could give a lot more chemotherapy, and when their bone marrows crashed, as they would do, we could just give them bone marrow transplant, and we could get more cancer cells killed and better outcomes for patients. Well, it turns out that there wasn't a whole lot of data for that, but back in those days it was felt that more was better so long as we could keep the patients alive. So they decided at that point to do a randomized study, and it was kind of like they thought, okay, for sure this makes sense. Of course we're going to show that more is better, because if a little bit is good, more has to be better. So let's do some randomized studies just to prove our point, and then we can move on. Insurance companies were reimbursing for stem cell transplants for patients with breast cancer, for instance. Well, there were 5 randomized studies that were done. 4 of them were reported, and they were completely negative. There was no improvement in outcome when you got the transplant compared with just conventional chemotherapy. A 5th one, however, was strikingly positive. In this one, you can see here, was by Bezwoda et al., published in the Journal of Clinical Oncology in 1995. You can see a complete response rate of 51% if you got the stem cell transplant versus 4% if you got just the chemotherapy. Now, complete response rate of 51% was unprecedented. And there was all kinds of significant interest in what was going on in this study. Why was it so positive in this study versus the other studies? And there were clear differences in what was given and how it was done. And so they decided they were going to study this in depth. And so it turns out Dr. Bezwoda was in South Africa and had done his studies there. And you can see here, this is the actual trial that was published in the Journal of Clinical Oncology. But a group of oncologists got together and said, okay, we got to get to the bottom of this. What is going on between Dr. Bezwoda's study? What can we learn from this phenomenal things that he's done there that we could potentially translate into clinical trials here, that we can really take what he's giving to these patients and see these same type of responses here in the U.S. So what they did was that they went, actually physically flew there. Actually, initially they asked for him to send documentation of the different patients and the case report forms, and he sent a few forms, but it really wasn't enough. And so then they decided that they were going to go actually to South Africa and go to his clinic and actually look at all the paperwork themselves and see just exactly how he did this. Well, to make a long story short, what they did was they reviewed all the literature that he had, the patient charts, all the case report forms, and there was a lot of disparity between what was reported in the paper and what they actually saw. They saw that they couldn't find consent forms for the vast majority of patients treated with the standard of care chemotherapy, for instance. And they couldn't even find patient records about some of these patients. There was no evidence that those patients had been treated. In fact, there was no evidence that there was a clinical trial approved by the regulatory authorities there in South Africa. And it appeared that he was just treating patients in his clinic with regimens, and there was really no standard protocol for that. So based on this data that came out in Lancet here in 2000, you saw that the original paper was retracted in 2001. But remember, the original paper was published, as you can see here, in 1995. So between 1995 and 2001, there were many hundreds of patients that were treated with, that had breast cancer, that were treated with high-dose chemotherapy with stem cell rescue, and to see if those patients could benefit, when clearly, if he had never made up this data and had it published in 1995, these patients would have been spared significant side effects. So why does this matter? As we mentioned, hematopoietic stem cell transplant is very costly, both in terms of actual physical cost and ICU stays and antibiotics, and they get very sick because their counts are so low for so long, but also in terms of substantial side effects with graft versus host disease, et cetera. And the treatment-related mortality is significant, depending on the treatment center, somewhere between 10 and 20%, especially at that time. Another very sad case is the case of the mumps, measles, and rubella vaccine. So we're going away from cancer now into the general patient population, and this affects mostly young children. So in 1998, there was a paper by Wakefield that linked the MMR vaccine, the mumps, measles, and rubella vaccine, to this syndrome that was associated with autism and diarrhea. And this syndrome he highlighted in 12 patients, and this was published in Lancet. And multiple groups looked at this data, and they couldn't replicate the findings. They looked at many other patients, and indeed, many of the big medical societies and pediatric societies came out saying that really we have to continue to vaccinate against MMR, but there was significant controversy regarding this. It was interesting that a reporter, whose name was Mr. Deere, uncovered multiple inconsistencies with the Wakefield manuscript. So he went back through, and actually is interesting, this was published in British Medical Journal, and he went back through and he identified each of these cases. He went back and contacted each of these families and asked them and tried to look for documentation of all of the things that were in Wakefield's paper. And there were three items to this syndrome that he had. There was regressive autism, nonspecific colitis, and the fact that these first symptoms appeared days after this MMR vaccine. So it had to have one or both of these, actually had to have both of these, and it had to occur right after the MMR vaccine. And what he found was that while there were a few cases of this, there's only one case of regressive autism actually in all of these, with several question marks, despite the fact that the Lancet article reported all of the first 7 of these. And the nonspecific colitis, only 3 out of 12 could they get any evidence of nonspecific colitis or diarrhea. And the symptoms happening after the onset, it didn't happen after the MMR or immediately after the MMR in any of the cases that he could document. So none of the 12 patients had this, and this was considered to be scientific misconduct, and indeed this was later retracted. Interestingly, Wakefield failed to disclose that he was working on a lawsuit against the manufacturer of the MMR and was paid over £400,000, pretty tidy sum of money. And so he was stripped of his license, and the Lancet paper was retracted in 2010. But remember, the Lancet paper was first came out in 1998, so this was 12 years later. 12 years where many people stopped giving their children the MMR vaccine. And indeed, what you see here is here it came out in 1998. You can see that the proportion of people in England and Wales that had the MMR vaccine among children dropped substantially from the mid-90s, and you need, for herd immunity, you need to have over 90% vaccination with this, and it dropped down to 80%, and you can see there's a big spike in the number of cases of measles. And indeed, the same thing held true here in the U.S., where there has been spikes of measles, whereas before it was pretty under control. Another case that I want to share with you is a clinician, Eric Pullman, who's involved in several different clinical trials. He admitted to falsification and fabrication of clinical results. So he only admitted to that after one of his staff came forward and reported inconsistencies to management. One of his studies was a longitudinal menopause study. So this reported on 35 women studied at baseline and 3 years later. Turns out that when the data was analyzed, all but 3 of these patients had fabricated and falsified data. This was published in the Annals of Medicine and 6 other academic journals and used for 9 different NIH and 2 USDA grant applications. Now, he didn't get all of those grants, but he had another study, a longitudinal study of aging. He liked these longitudinal studies. He exaggerated the number of patients tested. He changed values to create a trend in the aging process. And he presented this data in 3 different grant applications. And then he had a prospective hormone replacement study. Now, this one was one of the silliest ones, I think, here, because this was a double-blinded study. And yet, he presented preliminary data reportedly from this study when he did not have access to the unblinding. So he said, well, this group is doing much better in here, and here's the data to support that, but the data was still blinded. So one of the reviewers said, hey, something's not right here. So he'd applied for $11.6 million in funding, and he was awarded $2.9 million of grants. From 2000 to 2004, the University of Vermont and the NIH investigated him, and they found that he'd presented false testimony in documents and had influenced witnesses, trying to tell them to say things that weren't true. He eventually admitted to the destruction of electronic evidence and of his falsifications and fabrications. So he had to go to jail for a year, 366 days, actually a year and a day, and he lost any access to any future grants, and he also was barred for life, from receiving any funding from any federal agency, and also from the federal health care program. So no Medicare and Medicaid for him. So here was his defense. His defense was, well, I misrepresented minor points to ensure that a grant was refunded. Not good enough. Well, my staff, they were dependent on the grant for their salaries, and they have families. Can't do that. And he wanted to be recognized as an important contributor to the field. Well, egos do get in the way, but just remember, this is not good. Here's another interesting example of somebody that is high up, was a professor at Harvard. He was in the psychology department there, and he was the director of Harvard's Cognitive Evolution Laboratory. Sounds really cool. Nice title. Interesting guy. Turns out that he was doing experiments where they were playing, they had videoed monkeys giving cues following an auditory signal. And what they did was they marked whether a monkey responded to the cue or had no response to a verbal cue. And he typically would grade this with one of his lab personnel. Well, he happened to not be there, the videos were ready to go, and there was 2 laboratory personnel, and they graded it, and they saw no association. He said, no, that can't be, and he went back and he changed the remarks of the lab personnel, and he said, no, it should be this way, clearly there's difference here. Fortunately for the lab personnel, they stuck up for themselves. They said, no, this is not right. I do not agree, and I will not sign off on this, and I'm going to take this to the next level. They did, and after some significant back and forth, there was an investigation initiated in 2010, and they actually found that he was guilty of misconduct, and the case was turned over to the Office of Research Integrity here at the NIH. Interestingly, at that time, he was working on another book called Evolicious: Why We Evolved a Taste for Being Bad. Certainly, he seemed to have evolved that taste. And finally, in July of 2011, he sent a letter to the dean of the Harvard University saying, I've decided to pursue other areas and I'm going to be leaving the university at this time. Not even mentioning the ongoing controversy surrounding his scientific misconduct. So, what is the fallout from all of these cases of scientific misconduct we talked about and other cases of scientific misconduct? Well, certainly it impacts other scientists. They can become quite upset because they've seen this data, they've tried to replicate this data, or they've tried to understand it, or they've used this data to base other experiments on. This, without truth, we can't move forward. We have to have a basis for moving forward, and that stepping stone, that basis, has to be firm. Institutional reputations are damaged. So I don't think that patients are going to be wanting to go to South Africa for their transplant. And entire disciplines may be tarnished, such as stem cell transplant for solid tumors. There may be good opportunities in the future, and there may be other ways to do this, but it may be that a whole generation of academics will say, oh, no, that doesn't work, that's tainted now. Perhaps even more importantly, in the wider audience, patients and their families, they look to us to bring truth about science and to bring truth about what's going on, especially in the medical sciences when we're impacting directly their care. And they want to know why the promising results that they heard just a few years ago no longer are valid. And so they may lose their trust in research and in what we are doing. Certainly the public relies on our trust, and it could undermine science in general. So how is research misconduct handled at the NIH? Well, certainly here we have the NIH Agency Intramural Research Integrity Officer, or REO. And if you have a allegation of scientific misconduct, typically what you would do is you would go to your supervisor, and that would go up the chain of command and make it if there is something potentially going on, make it to this office. What they do first is an assessment to determine if the allegation meets the definition of scientific misconduct. And then they form an inquiry committee. Now the inquiry committee goes in and actually looks at the evidence, and they come up with a preliminary decision of whether or not a full investigation is needed. So the inquiry is just to get all the data together to say, yeah, it looks like we need to look into this in more detail, or no, actually, this can be easily answered. This was just sloppy data capture or something like that. So if an investigation is warranted, the investigation committee will determine if scientific misconduct has occurred, and then the ARILO, which is the NIH Agency Research Integrity Liaison Officer, concurs and can impose sanctions on that individual. So why would somebody manipulate data? And these are just a few points. Sometimes they just want to make their own data look more convincing. Sometimes they want to get something that can support their specific hypothesis. So some people will say, well, obviously this has to be what's going on, so I don't really have time to do the experiment, but I'm just going to throw up a couple of numbers on a graph, make it look good, and say, you know, this is what's going on here. Not good, not okay. So they need an experiment to finish the manuscript. They just run out of time, and we're behind. There's another lab that's about to publish. We don't want to get scooped, so let's just throw in this data here. I'm sure this is how it's going to turn out. We'll do the actual experiment. We'll swap it when the paper comes for review. We can just say, oh, well, we got another set of data, and it looks better. Again, if you submit it, it's already there. If you write it in your lab book, you've already committed potentially scientific misconduct. Or you're running out of time, you didn't plan ahead, and I've got to give a talk the next day. Let me just throw something together here, and if it's not true, I just won't use it in my next talk. Well, that's not okay to do. We have to be clear on what is truth and what is not, and we shouldn't be reporting or showing data that is not true. So remember, those who are caught manipulating data could be barred from life from ever receiving government financial support for research, or even be sentenced to jail. This is another case, before we talk about plagiarism, this is another case that was just reported last year, where this Anil Patti at Duke had done a lot of different data with genetic analysis, and was trying to come up with personalized testing, and this was going to be the wave of the future, how he could figure out from genetic testing who was going to be responding to different agents, made big progress, got a lot of big grants, and turns out that much of the statistical assumptions that he was making were actually not good. They were misrepresenting the facts, and in fact this led to at least 10 papers being retracted, and Duke University had to give back to the American Cancer Society over $700,000 in grants that had come in as a result of Dr. Patti's research, and he had to leave Duke. All right. let's talk a little bit about plagiarism. We've talked mostly about fabrication and falsification up until now. Plagiarism is a little bit separate and probably, you think, a little bit more straightforward. But really, it's the appropriation of other person's ideas, processes, results, or words without giving the appropriate credit. And you can see here, she's writing something here and it's copying it over here. That's not really good to do. And he's copying her also. So there's duplication, co-submission, and plagiarism. Those are three separate groups that are often, words that are often put under plagiarism. So what is duplication? This is actually potentially legitimate. So you can give clinical trial updates. So you may report on the first 14 patients, you may report of the trial, you may report some safety data, and then after you've enrolled all the patients on the trial, then you submit a paper and it has 28 patients, and now you have all the safety data for the whole trial. There is some duplication because you did initially report the first 14 patients, but this is not considered plagiarism. Meeting proceedings. Sometimes you can have things that are in abstract form in a meeting, and then you can later publish them in the full manuscript at a later time. Again, that could be duplication, but it is not plagiarism. And errata. Sometimes you have a table that maybe something got transposed, or you find an error later and you send back in the full table with the fixed numbers, the numbers that were transposed or whatever. Again, some of those numbers in there are duplicated, but so long as you have the link to the right article, that is not plagiarism, that is duplication. That is perfectly fine. Illegitimate, of course, if you duplicate and it's plagiarism or co-submission. So there's, and co-submission basically is just submitting the same thing to two different journals and they both get published. You really should only submit a manuscript to one journal, and then if it's rejected from there, then submit it to another journal. So there are multiple different plagiarism detection methods. I'm not going to go through in detail, but I will tell you that these plagiarism detection methods are used by all of the journals now, and they will often, I review for multiple different journals, and they will often send me, as editor in some of the journals that I edit, they'll send me a paper and they'll say, this has a 24% similarity with another article, and I'll look through it and say, does this meet plagiarism or not? Sometimes it's just the methods section that is very similar to another methods section, or it's words that are, short phrases that are used. But if it's an entire paragraph that's used, then it rises to plagiarism. So there are plagiarism tools. This is on the ORI website. You can see ori.hhs.gov, plagiarism tools. You can go on to this website and you can look. Deja Vu is one of these, and includes a database of over 70,000 pairs of citation and notes. And this is a common way of checking, is there anything that's taken in this manuscript that could be potentially plagiarism, especially if you have co-authors and you're not sure, how did that postdoc do this phenomenal job here with this? Did he get it from somewhere else? So again, this is the Deja Vu I mentioned in the previous slide, and you can see a website here, how to get to it. So here is an interesting graph that was published in Nature several years ago, and you can see here over time the citations to Medline. You can see rising up to 600,000 citations here. and then the number of duplicates per 1,000 citations, and that's on this axis. So it starts out at 2 duplicates per 1,000 citations, and you can see here it starts to maybe go up. Maybe that's because we're better in annotating and finding these, but it's something that there certainly are a finite number of cases of this, and it's something that can be easily tracked now a lot better than previously. And you might say, okay, you talked about all those fabrication and lots going on in Asia or South Africa. That doesn't really impact us here, but there were those cases here also. It turns out that if you look the suspected duplicates and the relative contribution to Medline, there's really no difference whether you're, based on where you come from. Still, most of the data that gets published in Medline comes from the US, and most of the suspected duplicates come from the US. Okay, going to have a few questions here. Some of these they're very easy, and some of them could be grounds for more debate. So I'm going to ask a series of questions that go over if authorship is appropriate. And I'm going to ask for you to answer. So is authorship appropriate if you wrote the paper? Okay, surprisingly easy, right? Okay, yes. How about if you grew the cells used in the study that was published in the paper? Thoughts about that? I see somebody saying no. Anybody want to counter? Okay, let's see what I wrote. Yeah, so if you just give the cells, no, it probably doesn't rise to that level of authorship unless you made an intellectual contribution to this study. Probably not going to get you on the paper if that's all you did. Okay, how about if you performed half of the experiments in the study? Yes, okay, I heard a yes. Thank you. Yeah, so if you performed, obviously, then you should get it. How about if you identified and ordered the commercial supplies used in this project? Right, exactly. No, that does not rise to authorship. Okay, how about this? What if your girlfriend worked on the project? Okay. Yeah, you guys think that's crazy too. Yeah, no. So how about if it's your boss and he didn't work on the project? How about if it's your branch chief or your lab chief? If they didn't work on the project, if they didn't provide any intellectual contribution to the project, then no, they should not be listed on there. How about if you thought up the idea for a project? Well, I think this might be a good project, and somebody else takes it and runs with it. What do you think about that? So, speak up. So the response was, no, if you just had an idea and didn't do anything about it, you shouldn't be an author. And I see you have differing opinion? So great. So yes. So if you did, so this probably is a difficult question. And if you conceive the basic hypothesis, maybe you should get credit. But especially if you worked with them to help develop the trial design. But if you just casually mentioned it to somebody, kind of walking down the hallway and said, "Oh, wouldn't it be cool if we did this?" and they took it and ran with it, and you didn't have any other contribution, then maybe not. But if it was in your area and they came back to you and they said, should we do this, should we do that, maybe so. So again, some of these things are situational. Some of these things, you'll know it kind of when you see it. But remember, authorship is a privilege, not a right. You must have made a meaningful contribution to a manuscript to earn it. And I would say, if in doubt, at the beginning of a project, if you're working on a project and you think you should be an author, sit down and talk with the other people involved with that and say, hey, listen, I think I should, what about the authorship here? Could I be part of the authorship? Because I think that I want to do this, I want to make this contribution. And, if it's a clinical trial, sometimes just sending in patients for a clinical trial may not be enough to warrant authorship on a study. But maybe if you are there and you are seeing the patients and you are making treatment decisions and you're writing them down and you're helping craft the manuscript, obviously that does. But maybe just seeing the patients or just administering the drug and following a protocol, maybe that doesn't. So you should discuss these things beforehand, because it's a lot better to figure this out up front. So the following by themselves are not criteria for authorship. So as we mentioned, holding a position of administrative leadership, the branch chief, the lab chief, just because they're the branch chief or lab chief doesn't mean they should be on the paper. Contributing patients or reagents, we kind of talked about that also. And collecting and assembling data. Just because you are going through the process of getting the data, putting it into a spreadsheet for somebody else to interpret, that doesn't mean that you made a contribution. However, if you are the one interpreting the data and coming up with maybe this correlates with that, then maybe that does, and maybe that turns into a table or a figure in the paper, maybe that does warrant authorship. So, as I mentioned before, the best time to address the authorship is at the beginning of a project, not at the end. If you're at the end of a project and there is a dispute, most of the time you can discuss with the other people and you can come up with an agreement over who should be on a paper. The most common types of authorship disputes that I have seen have been about who should and shouldn't be on the paper, but even more common than that, who should be the first author and who should be the senior author. Those are the more common types of disputes that you get. Besides direct dialogue with the members, which probably 85% of the time works, if that doesn't work, then you can go for mediation. There is an NIH ombudsman who can help mediate these types of disputes, hear the sides, get them to understand and be reasonable with each other. But if you still can't get to the bottom of it, then you can go to this peer panel, which is basically a voluntary 3 or 5 member panel. It has to be an odd number so that you can't have a tie. Member that have expertise in the area that is under consideration, and they then have a binding decision, and they might decide against you or they might decide for you, but then that's it. Or you can go directly to the scientific director if all the authors within one institute, or you can go to the deputy director of the intramural research program if they are in different institutes for a binding decision there. You want to start off with the top and then work your way down, because you don't want to go to the atomic, you don't want to push that button too many times, otherwise they're going to say, what's going on with this person? It keeps coming back to us. This may be a problem person. Okay, last little bit here, we're going to go back through about the fabrication and falsification. And we're going to assign which of these cases that we talked about were fabrication and which were falsification. So first of all, this South Korean researcher who had the cloning of the stem cells, was that fabrication or falsification? Falsification? Did you say? Or fabrication? Fabrication? Fabrication, yes. Anybody else? Both. So so fabrication, 9 out of 11 cell lines were absolutely just fabricated. And there was image manipulation, so that's falsification there too. How about Bezwoda and the breast transplants, the bone marrow transplants in the breast cancer patients? Falsification, fabrication, both? Falsification for sure, and I heard fabrication, so let's go with both. Yeah, so Bezwoda made up patients. That's at least part of his fabrication, and stated that he had consent for all of the patients when, in fact, they couldn't find consent forms for anybody. There was not even a consent that had been reviewed by the IRB. Okay, how about Wakefield and the measles vaccine? Fabrication or falsification, or both? Falsification, yeah. Fabrication, yes, no? So he actually made up the colitis on these patients. As you saw, there was no one that had colitis that could be documented. And the date of reaction was falsification. So some of the ones that he said happened afterwards, they actually happened before they got the MMR. So they couldn't be due to the MMR. Pullman and his longitudinal studies. Fabrication, falsification. You guys are seeing a trend here, aren't you? Yeah, so he made up patients, completely made up the data for those patients, and he destroyed data. So falsification, remember, falsification is changing the data in any way, including destruction of data. So you shouldn't be destroying data if you're destroying it to try and cover your tracks. That's actually falsification. And Hauser in his monkey studies. Falsification and fabrication? Yep, absolutely. So he actually fabricated the sounds that the monkeys heard and falsified the monkey responses. So in summary, really what I wanted to share with you is that the ethical conduct of science is each of our personal responsibility, that we must not commit falsification or fabrication of data or plagiarize the work of others, and that performing scientific research as a career is a privilege. By performing it in an ethical manner, you will maintain the highest standards of scientific integrity. Thank you, and I'd be happy to answer any questions you might have. Any questions? Yes?
Is it possible to have plagiarized something unintentionally just because of background knowledge or familiarity?
Give me an example of what you're thinking about. So about ideas or the exact wording of somebody else?
Concepts or ideas or general knowledge, you might end up trying to claim general knowledge.
Right. So general knowledge that has been out there for a long time, typically this is not, if it can be seen in multiple different sources, typically this is not something that you have to attribute. But if there's only one or two reports of this previously, you should reference that in your, say, your background section. You share that idea and then you reference where that idea came from. That is the best way to do that. However, if you I think an idea would be if you came in with an idea that somebody else had previously come up with, and you didn't want to give that person credit for that idea, and you made it seem like it was your own idea, then I think that would be potentially plagiarism. So I think in general, you try and be generous when you're going through your background, and if something came up as a result, if you are writing this down, if you came up with this as a result of your search of the literature, then I would just reference the key sources where that came from. Other questions? Okay, thank you very much.